The last part of the process is what goes on in the eyes of the observer. We refer to this as the response. And like everything else, the response is a function of the wavelength lambda. In fact, the response can be written this way. It is the integral over wavelengths of the spectrum of the incoming light, of the reflectance, and of the response of the eye to light at that particular wavelength. So what we sense in the photoreceptor cells in the back of our eye is essentially the result of this particular integral. Let's see how that works. Here is a cutaway diagram of the human eye. At the back of the eye we have the retina. If we're looking at the surface of the retina, we see a lot of blood vessels and we can see the optic nerve, which is what connects the eye to our brain. If we take a cross-section through the retina, we make a slice through there, we look downwards, we see that there are layers of nerves and other sorts of cells, and at the bottom of this stack we have the light-sensitive cells, and they look something like this. So what's quite surprising is that the light-sensitive cells are not on the surface of the retina. They're actually about 500 micrometers below the surface of the retina. So the light that falls on the retina has to fight its way down through blood vessels and nerve cells and all sorts of things to get to the light-sensitive rod and cone cells. Let's look at the rod cells. They're these small worm-shaped objects that we see in the back of the retina. They contain a chemical called rhodopsin, and is quite an important chemical because when a photon of light hits it, it changes its shape. It changes its shape from this to that, and we can see that one arm of the molecule has in fact been spun around by the impact of the photon, and that is eventually converted into an electrical signal which travels along the optical nerve to the visual cortex of your brain. The rod cells are very, very sensitive to light, so these are the cells we use in low light conditions. This is what we use at night. And the way they're wired makes them also very sensitive to motion. That's what we use to determine that something's moving in the edge of our field of view. The rod cells are sensitive to a range of electromagnetic radiation, as shown by this graph here. So this is the response curve of the rod cells, and we denote it by the symbol m, and it of course is a function of wavelength lambda. The rod cells are maximally responsive to light at 498 nanometers, basically mid-green color. The other very important class of cells in the human eye are the cone cells, and one of them is indicated here. They're not that conical, they're certainly fatter at the bottom than they are at the top, quite a different shape to the rod cells. The cone cells contain chemicals called photopsins. They're related to rhodopsin that we have in the rod cells. There are different flavors of photopsins that are sensitive to light at different colors. The cone cells are what we use for our everyday daytime activities. In the daytime we use our cone cells, not the rod cells. The cone cells contain a chemical called photopsin. And photopsin is related to rhodopsin, but there are three different flavors of photopsins. The photopsins that are sensitive to red, to green, and to blue light. And so there are three different types of cones, those that contain these different types of photopsin, sensitive to red, green, and blue. So we say that human beings are trichromats. It means we have got cells that can sense three different colors, sensitive to different bands within the spectrum. And that's what's shown in the graph over here. Often these bands are referred to by the letters S, M, and L, which stand for short wavelength, medium wavelength, and long wavelength. We'd also refer to them as blue, green, and red in that order. The response of the short, medium, and long cone cells are shown here. And we can see that there's quite some overlap, particularly between the medium and long wavelength cone cells. And also shown here for reference is the response of the rod cells we looked at in the previous slide. Let's look more closely at the retina, and there are two key landmarks in the retina. One is what we call the fovea, where we have an enormous concentration of light-sensitive cells, and the other big landmark, which is where the optic nerve joins the retina, and that area has almost no light-sensitive cells. It's actually a blind spot in your eye. So if we project those 2 points down and we draw an axis, we can plot the concentration, the density of cone cells as a function of angle away from the fovea. So on one side we have the direction towards your ear, on the other side we have the direction towards your nose. And we can see there's an enormous concentration of cones in the foveal area and a much lower concentration of cone cells in the rest of your eye, what we call the peripheral vision area. If we look now at the distribution of rod cells, we see almost the converse. We see that there are very many rod cells in the peripheral part of your vision, not too many of them at all in your fovea. So the fovea, we have a very high concentration of color sensitive cells. The rest of the eye, there are a large number of cells that are sensitive to light but don't distinguish between different colors.